So, we are here in this podcast, Mitogenesis, rewiring life from the inside. So, welcome to Mitogenesis, where we dive deep into the hidden engines of life. Today's episode is all about remarkable relationship between stem cells and mitochondria, a partnership that defines how cells grow, specialize, and even rejuvenate. Let's start with the basics. Stem cells, whether embryonic or induced pluripotent, are the raw material of regenerative biology. But what most people don't realize is that these cells are metabolically immature. Their mitochondria are small, rounded, sparse, and functionally minimal. Why? Because pluripotent cells rely heavily on aerobic glycolysis, not oxidative phosphorylation. It's a metabolic setup that prioritizes speed and flexibility over power and endurance. Think of glycolysis as sprint, fast but not terribly efficient, and it suits the chaotic, rapidly developing nature of early development. Here's where it gets fascinating. As stem cells start to differentiate, to commit to a lineage, whether it's neural, cardiac, or hematopoietic, their metabolism shifts. This isn't just a flick of metabolic switch, it's an orchestrated transformation in mitochondrial form, function, and behavior. And that's where our guest today steps in. So we have Vasudha. Basudha Banerjee is an enthusiast of regenerative medicine. She's very young, but she's already showing a potential in stem cell biology in regenerative medicine. And we've already had a great discussion with her as an expert on mitochondria. Welcome, Basudha, to this conversation.
Thank you so much, Dr. Khurana. So, indeed, what we are discovering is that mitochondrial dynamics, the processes of fission and fusion, they are not just the side effects of differentiation, but rather they're the drivers. Now, as we're talking about all the stem cells, so pluripotent stem cells, as they begin to specialize, we see a dramatic increase in the mitochondrial biogenesis. The mitochondria elongates, their cristae matures, and they begin to assemble into a fully functional electron transport chain. That metabolic sprint becomes a long distance endurance engine powered by oxidative phosphorylation. In fact, you can predict the cell's developmental fate by watching just its mitochondria. It's like reading the soul of a cell through its metabolic pulse. And here's the kicker. When we try to reprogram somatic cells like skin cells back into induced pluripotent stem cells, we see a reverse mitochondrial reset. The mature networked mitochondrial regress. They fragment, their oxidative capacity drops, and the cell reverts back to glycolysis. So, this means that mitochondria are not just passengers, they are the architects of cell fate. They're either building towards specialization or helping wipe the slate cleaning during the reprogramming.
Well, that's an incredibly powerful insight. Now, let's take this to clinic. When we talk about stem cell transplantation, whether for neurodegenerative diseases, muscle repair, or immunotherapy, one of the least appreciated but critical variables is mitochondrial health. If we transplant stem cells with underperforming or stressed mitochondria, the grafts fail to integrate or survive. That's where mitotropic optimization comes in, a term still catching on in the field. It refers to preconditioning stem cells before transplantation, ensuring their mitochondria are in a top form. This could include redox priming, NAD plus enhancement, mitophagy tuning, and even the use of mitochondrial transfer technologies. In the future, we may personalize stem cell therapies not just by matching immune markers, but by mapping mitochondrial readiness. And that's where AI will likely play a key role, predicting which mitochondrial profiles give the best outcomes in specific tissues.
That's very interesting. So, imagine an AI system that analyzes the mitochondrial morphology, the ATP output, redox state, and the fusion fission balance in real time. It is offering a quality control dashboard for the stem cell production lines. We are not yet there yet, but the groundwork is still being laid. Ultimately, the mitochondria may be the quality gatekeepers of the regenerative medicine, and understanding their role in stem cell fate gives us the power to not only grow cells, but to guide them with precision, resilience, and purpose.
Well, thank you for that incredible insight. To our listeners, if you're building stem cell platforms, working on cell based therapies, or just fascinated by hidden gears of biology, remember this: no cell therapy is complete without a mitochondrial lens. Until the next time, keep decoding life from inside. This is Mitogenesis. Thank you very much. Thank you, Basudha.
Thank you so much.